Now, we wanted to look to see if these structures had hallmarks of other extracellular traps, including typical proteins, including histones, myeloperoxidase, and neutrophil elastase. And I'm going to turn down the lights to make it just a little bit easier to see. And so what we've stained for is in blue is Hoyx, which is a condensed chromatin stain, normally used to stain nucleus, but in areas where the extracellular traps are thicker, you'll also see some staining. That's Cytox green, that DNA intercalating dye, and then specific antibodies for these different proteins. And what I hope you can appreciate, especially in the merge, is that these areas where we have these extensions coming off of the cells, we have co-localization of that red staining in that same location, suggesting these proteins are part of the extracellular trap. Now, to make sure that this staining is specific, we did staining controls using either no primary antibody or a secondary isotype control, and we don't see any of that red staining. The next question was, well, these structures seem to be extracellular traps. Can they actually kill bacteria? And so to find this out, we did a killing assay where I set up two parallel systems. In one, we're going to infect GBS, or macrophages with GBS, and they'd be expected to release their extracellular traps. In the second, we're going to leave DNase in the system to degrade those structures. And again, at 1 hour of infection at the MOI of 20 to 1, when we count the colonies, at the end, we see at those wells treated with DNase, we see a significant increase in the number of bacteria recovered, suggesting that if we disarm the extracellular traps with that DNase and release that high concentration of those proteases, we lose our antimicrobial effects. We've also done live dead bacterial staining. So both of these, cyto9 and PI, or propydium iodide, are DNase binding dyes. Propydium iodide is restricted from live cells, meaning it only gets into dead cells and will quench the green signal. So what we see here is a bunch of live bacteria staining green, and then in red we have the trapped fibers that you can see and these dead bacteria that are lighting up red. When we look at the overlay, what I hope you can appreciate is those cells that seem to be dying seem most closely related as far as distance to the extracellular trap fibers, where you can have single dead bacterial cells within a chain of live ones that seem to be associated with those extracellular traps, suggesting that there is some degree of bactericidal activity there. We also wanted to look to see how specific this response was in our system. Are these only from these tissue differentiated macrophages? Is this strain specific or bacterial species specific? And so if we infect either with a different strain of GBS from a different capsular type or E. coli cells, we see a similar response from these cells, suggesting that it's not strain or species specific. We also used heat kill E. coli or GBS, and we saw similar responses. If we look at a different type of macrophage, monocyte-like cell line, the THV1 cells that we PMA treat to make them macrophage-like cells, we see similar responses. So again, compared to uninfected cells, those that we've infected with GBS, we see a significant increase in the amount of extracellular traps released, like here. If we treat with DNase, we lose those structures. Now, we expanded our conditions a little bit to see if bacterial cells were important for this process. So I used sterile filtered supernatant, and when we did that, we see no response compared to uninfected cells, suggesting that bacterial cells, that interaction seems to be driving this response. If we look at two inhibitors of the cellular cytoskeleton, they're important for cellular shape and potentially important for release of these extracellular traps. If we pre-treat the cells with cytoclasin D that inhibits the actin pluralization and actin cytoskeletal, we see an inhibition of the extracellular trap release compared to those untreated infected cells. If we treat with the cortisol, which inhibits microtubules, we see no response compared to the uninfected cells. So together, it seems that GBS cells are important for this response, and actin polymerization may also play a role. Now, this is a complicated slide, but the key point is that reactive oxygen species are thought to be important for this extracellular trap response. So regardless of the type of stimulation a neutrophil, in this case, receives, the big thing is that all of these seem to lead to the generation of reactive oxygen species. And it's thought that the process of releasing these extracellular structures is dependent on ROS that then breaks down granular and the nuclear envelopes, allow mixing of proteins like myeloperoxidase and elastase with the DNA so that they can be shot out of the cell. So I wanted to look to see, is reactive oxygen species, or ROS, important in placental macrophages to release these traps? So again, using the same infection setup at MOI of 21 for 1 hour, but now we're going to pre-treat these cells with DPI, which is a non-specific inhibitor of NADPH oxidase, and then cytoclasmin D again, that actin cytoskeleton inhibitor. And we can see, compared to those cells that we've infected with GBS, if we pre-treat with either DPI or cytoclasmin D, we lose this extracellular trap response. We don't see as many of those structures coming off cells. Now, if we leave the cells uninfected but treat them with PMA, protein kinase C agonist that would be likely to drive ROS production, we see a similar response at 1 hour to GBS infection. So let's narrow this down a little bit more. So I'm using a fluorescent dye that when it reacts with reactive oxygen species becomes fluorescent. So these are live cell infections. On the top we have staining in blue for the nuclei, in green for our extracellular traps, and then on the bottom just the ROS production so you can see it independently. Compared to those uninfected cells that regenerate a little bit of reactive oxygen species, GBS infection and PMA treatment drives a lot of ROS production in these cells. We see very little in both DPI and cytoclasmin D. And if we actually calculate this into an index that takes into account the total fluorescence, the number of macrophages that are there, and those that are making ROS, we can see that GBS infection and PMA treatment drive ROS production up, and if we pre-treat with cytoclasmin D and DPI, we lose that response. If we put that right next to our extracellular trap quantification, we see a similar trend, suggesting that reactive oxygen species are necessary in this case for the release of extracellular traps. So what's going on here? What's the actin cytoskeleton have to do with the release of reactive oxygen species? And what we think is happening, though we haven't confirmed this, is that the NADPH oxidase complex actually consists of several subunits, 2 of which interact with the actin cytoskeleton. So by pre-treating with cytoclasmin D, we may actually be inhibiting that NADPH oxidase complex from forming. So thinking back to what we talked about what macrophages are doing normally during pregnancy, a lot of their role, especially in the 2nd and 3rd trimester, is involved in tissue remodeling. And so we've already shown that some proteases, like neutrophil elastase, get into these extracellular traps. I wanted to look at other metalloproteases, extracellular matrix digesting enzymes that may also be present. And so we looked at several different MMPs, MMP 1, 7, 8, 9, and 12. And again, using similar staining and immunofluorescence, we can see that these metalloproteases are also present within these extracellular trap structures. Not only are they in the structures, but if we look at the supernatants after just 1 hour of infection, we can see a significant increase in the amount of MMPs present. MMP 8 and MMP 9 are shown here. Now, a lot of these enzymes, because they're stored in an inactive form, because they break down other tissues, we wanted to see if they're active when they're released into this system. So we used a fluorescent substrate that is cleaved by multiple different MMPs. So this isn't specific for any of these specific MMPs, but just looks at general metalloprotease activity. And again, after just 1 hour of infection, we see about a 50% increase in the amount of MMP activity in this system. So why is this important? Many of these metalloproteases have been implicated in preterm pathologies, including preterm birth for MMP1, preeclampsia, and premature rupture of membranes for MMP7. MMP8 and 9 are actually being looked at as a biomarker for intrauterine infection because in amniotic fluid of women with confirmed intrauterine infection, these have been found to be elevated. And then MMP12 hasn't been found to be elevated in pathology, but is very important for spiral artery remodeling to set up that placenta and that nutrient exchange. Now, interestingly, this has been shown to be released by alveolar macrophages in the lung and may contribute to COPD pathology. So this all comes back to the structure of the fetal membranes themselves. And if we look at the diagram here, we can see that these two layers, the basement membrane and complex layer, are really full of collagens and extracellular matrix proteins. These enzymes would all be predicted to act upon that layer, potentially weaken or damage that structural layer that provides most of the tensile strength of this material, these tissues. So everything we've done so far has been in a plastic dish. And so we wanted to look, is this actually happening in vivo? Or is this just a artificial kind of response to the system that we're looking at? So we went back to our fetal membrane tissue model. And I used a staining strategy that has been used by others to identify extracellular traps within tissues. And so in blue, we have the nuclei. In green, we're staining for neutrophil elastase, which we've shown has actually been produced by macrophages as well. And then in red, we'll see histones. This is the choroidal surface. The amniotic epithelium would be over here. If we look at this in more detail, you can see there are several cells releasing neutrophil elastase into the surrounding tissues that co-localize with histones as well as DNA, that nuclear stain. And we merge them together. You can see that at the white arrows. Now, contrast this to what we see at the yellow arrow. We have a well-defined cell with well-defined granules and a well-defined nucleus. So this would be a normal macrophage that has not undergone the extracellular trap pathway to basically condense and mix all these contents together. Now, this staining strategy is not specific for macrophages. There's very few neutrophils within these tissues. But if we use a strategy that's more specific for macrophages, we can do this by immunohistochemistry, again, using our fetal tissues. The choroidal here's the amniotic epithelium. We can identify an area of GBS invasion into these tissues. And at that site, we can see CD163 positive macrophages present. And we can, in some sections, identify macrophages that seem to be releasing histones out into the extracellular space. If we look at this by immunofluorescence again, we can identify CD163 positive macrophages. And this time, we can see them dumping contents containing histones and MMP9, which we've shown are contents of the extracellular traps, again, into the extracellular space. You can see some red staining within the nuclei of other cells, and these tissues are making a lot of matrix metalloprotease 9. So we've shown that this extracellular trap response seems to happen within tissues. The relevance of that is still yet to be determined to some extent. These could have dual consequences of potentially trapping and killing microorganisms that are invading in these tissues, but you may also be causing localized membrane breakdown by the release of these proteolytic enzymes. So this is what's happening, it looks like, in placental macrophages, these tissue-differentiated macrophages. What do we know about macrophages in other systems? So we talked about the alveolar macrophages in the setting of COPD, release MMP12. Human glomerular macrophages seem to contribute to ANCA-associated